What's up, happy biopenguins? Today we're going to do something a little different. We're going to do a question from the CED. So this is going to model question number 5 on the exam is on meiosis and pedigrees. In humans, the genes that determines a particular condition has two alleles, one of which, a capital B, is completely dominant to the other, lowercase b. The phenotypes of 3 generations of a family with respect to the condition are shown in the pedigree in figure 1. Individuals are numbered. So anybody that's a square is a male, anybody that's a circle is a female. Shaded means that it has the condition, and unshaded means they do not have the condition. Part A always asks about biology. So Part A asks us to describe the process in eukaryotes that ensures the number of chromosomes will not double from parent to offspring when gametes fuse during fertilization. So why, when 2 gametes fuse together, do I have the exact same chromosome number as I did in the parent? Well, that actually has to do with meiosis. During meiosis 1, we see the homologous chromosomes will separate, leaving us with haploid cells. We then go through meiosis 2, and then the haploid cell again is going to split, and then we'll see the sister chromatid separating, which allows us to have a single chromatid of each chromosome in that one cell, so it's haploid. And then during fertilization, of course, that comes back together. So homologous pairs of chromosomes separate in meiosis 1. So the gametes are haploid, and each gamete receives only one member of each chromosome pair. Part B, explain how any one chromosome in individual 16 contains DNA that came from both individuals 1 and 2. So we have to think through what happens during fertilization as well as meiosis. So number 5 is going to be the offspring of 1 and 2. And so 1 was able to donate half this genetic material in its sperm, and then 2 was able to donate its half of its genetic material in its egg. And so 5 is going to actually have one homologous chromosome, well, one set of homologous chromosomes that came from 1, and one set of homologous chromosomes that came from number 2. And then during meiosis, we have crossing over where we see these non-sister chromatids. So say chromosome 1 is going to cross over. And so that's going to allow us to get a combination of both individual 1 and 2 on that chromosome that's coming from individual 5. So individual 5 inherited one member of each homologous pairs of chromosomes from individuals 1 and 2. During gamete formation, individual 5 crossing over occurred between non-sister chromatids in each homologous pair. Thus, each chromosome formed and passed on to individual 16 contains DNA from both 1 and 2. Part C. So in Part C, we're always going to modify some figure. We're going to be drawing a picture. We're going to be adding some words. Like there's just going to be some modification of a diagram. So use the template figure of the pedigree and the allele designations, capital B and lowercase b, to indicate the genotypes of individuals 2, 4, 8, and 6, 18. Okay. So what we have us to do is that we have to write 2 letters. These letters are going to signify whether they are homozygous dominant, heterozygous, or homozygous recessive for this. So we kind of have to look at the diagram and figure out whether we think that this is dominant or if we think that this is recessive, okay? So we look here at 3 and 4, I can see that I have 2 unaffected parents with an affected child, okay? And so that tells me that it's some type of recessive trait, okay? I also see that I have an affected male that has also an affected male offspring. If it was sex linked, this male would not be able to pass on that trait to its offspring. Okay, and so we believe that this is going to be autosomal recessive. And so in order for us to come with that, we're then able to look at number 2. 2 is able to pass on that trait to its offspring, which means that it must be heterozygous. Okay, if we look here, number 4 is also not affected, so it does have a capital B, and it also is able to pass on that trait, so it must also be heterozygous. When we look here, 8 is affected, which means they're homozygous recessive. And then this individual here, because of the fact they had a affected parent, we know they're also going to be heterozygous. So then part D, based on the pedigree, explain whether the inheritance pattern of the condition is sex-linked or autosomal and dominant or recessive, which we already explained this because of the fact that we were needing to know that information for the last question. And so if we look here, we can see that 3 and 4 are able to pass on that trait. So it tells us that it is recessive. And if I look here, I have number 7 and number 8. 8 is an affected female, 7 is an unaffected male. If it was sex-linked, there's no way you would have had an affected female down here because of the fact that this one had to have passed on that trait. So the disease phenotype is recessive and is autosomal, not sex-linked. It cannot be dominant because individual 3 and 4 do not have it, but their offspring 14 does. It is not sex-linked because if it was Y-linked, all male offspring of males with the disease phenotype would have that trait, and they do not. Hope that was helpful. Remember, 8 5 hey was just assessed by y'all.